Hello friends, today we are going to study about protein quaternary structure. So first of all, proteins are made up of subunits. So there are many proteins that are made up of subunits. Now say for example, this is protein 1 which has 4 subunits and a protein 2, maybe it has 2 subunits. Correct? Now these subunits are actually, we can also call it as a polypeptide subunit, because these subunits are a single polypeptide. So proteins are made up of subunits. Now these subunits, they play variety of functions like regulatory, catalysis, and or structural role. For example, ribosomes, it consists of a dozen of protein subunits. So based on these subunits, we can categorize protein into two categories. One is multimeric protein or multimers, and another one is oligomeric protein or oligomers. Now, as the name suggests, multimeric protein means the proteins that have multiple subunits. Oligomeric protein means the proteins that have definitely multiple subunits, but the number of subunit is 2 to 10. If it is more than 10, mainly, then only we call it as a multimeric proteins. Now, in this multimeric proteins also, say for example, this protein number 1, it has 4 subunits. So, when a protein constitutes of 4 subunits, in that case, the single subunit is known as protomer. So protomer is the single subunit of the protein if that protein consists of multiple subunits. Now these subunits can be identical also, non-identical also, but the common one is the identical one. And they can be symmetric also and asymmetric also, but the common is symmetric. The subunits can be non-identical also. For example, in hemoglobin, we'll see in the next slides that hemoglobin consists of two different subunits, but they are in symmetry with each other. The protein consists of multiple subunits, and each subunit, these are polypeptide. So say for example, a protein consists of 4 polypeptide subunits. Now, if we want to increase the size of the protein, it is better to increase the size of the protein by increasing its number of subunits rather than by increasing the length of polypeptide. Why so? Because if we increase the length of polypeptide, if the protein has, say for example, one subunit. So if we increase the length of the polypeptide, this subunit will become larger in size. Okay, but if we increase the size of the protein by increasing the number of subunits, because each subunit have an active site, so each subunit can perform functions and hence the efficiency of the protein will increase. But here it is only one subunit, so it will have only one active site. Doesn't matter how long we make the polypeptide or make the subunit, it is still a one subunit, and therefore it will still have a one active site only. So if we increase the number of subunits, so the number of active sites will increase and the protein can function more efficiently. Now the example of hemoglobin. Hemoglobin is the first oligomeric protein for which the 3D structure was determined. It consists of 4 polypeptide chains. That means it has 4 subunits. Each subunit is having heme group, which has iron in 2 plus form, and hence it can bind with oxygen. At a time, hemoglobin can carry 4 oxygen molecules. Okay. In the hemoglobin, apart from this heme part, the rest part is a protein. So the protein here is, it consists of 2 alpha chain and 2 beta chain. Alpha chains, the length is 141 amino acid residues, and beta chain, the length is 146 amino acid residues. And these subunits are arranged in symmetric pairs. That means each pair will have one alpha and one beta subunit. Hemoglobin can therefore be described as, what we can describe as that it is a tetramer. Why tetramer? Because it has 4 subunits, or we can say it is alpha beta dimer. This is one alpha beta part. This is second alpha beta part. So it is a dimer of alpha beta. The identical subunits are arranged in symmetric patterns. Now these patterns or these symmetries are of two types. One is rotational symmetry. Rotational means to rotate. That means when the subunits can be superimposed on one another by rotation around the rotational axis, it is known as rotational symmetry. It can be of following types. First is cyclic symmetry. Cyclic symmetry means when the rotation is about a single axis. When the subunits rotate around a single axis in such a way that after one rotation, they can superimpose each other. So that comes under cyclic symmetry. So it looks like this. This is a twofold and this is a threefold rotation, and they rotate around a single axis. That means around a single axis, if they rotate, they can superimpose each other. For example, hemoglobin. It has four subunits, 2 alpha and 2 beta. If they can rotate along this axis, this alpha can come and superimpose this beta can come and superimpose this. So by rotation around a single axis only, they can superimpose each other. So hemoglobin is a twofold symmetry. Now coming to dihedral symmetry, when the rotational axis intersects by rotating twofold, then we call it as a dihedral symmetry. So here you can see here this line is missing. So this is a twofold. This is one axis and this is also one axis. So this is a twofold symmetry. Here also the center axis and like this also there are axes. So that's why we call it as a dihedral symmetry. Next is icosahedral symmetry, when the rotation is around one or more of three rotational axes, then we call it as a icosahedral symmetry, which we generally found in viruses. So these are the type of rotational symmetry. Apart from rotational symmetry, one more kind of symmetry is helical symmetry. So helical symmetry means when subunits are superimposed by a helical rotation, and this we mainly see in viruses. Limits to the size of protein. How the size of the protein is limited. Who will decide that what should be the size of the protein? What should be the maximum limit for a size of a protein? Now let's take the example of a virus. The general structure of virus, we know that it has a capsid head, and inside that capsid head, they pack their genetic material. Now, because the space is very small, that means if the nucleic acid will become very large in size, they cannot able to pack it in this small head. Correct? Now, we know that the nucleotides present in nucleic acid, they only decide what will be the sequence of amino acids. And this will only decide the size of the protein. So if we want to increase the size of the protein by increasing the length of polypeptide chain, and as we know that the amino acid sequence in a polypeptide chain is because of the nucleic acid sequences. So if we need to, or if the universe want a long polypeptide, then that much long nucleic acid will also be needed to code for those amino acids. Correct. So it will be very difficult then for the virus, for example, here to fit that much large nucleic acid in its head. So it is easy for the virus to encapsulate a small size of nucleic acid. So small size of nucleic acid means if the size of the nucleic acid is this, for example, so this much gene suppose is responsible for the size of polypeptide chain of approximately this length. So it is easy if it makes multiple copies of this polypeptide. That means a protein with multiple subunit. Each subunit of small size but having multiple subunits. Correct? It is easy because in that case we need a small size of nucleic acid. But if we go for a large polypeptide chain, then in that case we will need a large size of nucleic acid. So it is always easy when anything is small in size. It is very handy. So nature also selects such proteins only. Therefore, it is common that the proteins will have a multiple subunits. If they have a multiple identical subunits, that means that small set of nucleic acid is only responsible for producing that polypeptide. So it is very easy to make multiple copies of that subunit to make a protein rather than to make a single large polypeptide chain of protein. Correct? So this is one reason that limits the size of a protein, that is genetic coding capacity of nucleic acid. Now, second is the accuracy of protein biosynthesis process. Though the process is very accurate, though the damage rate of protein biosynthesis, that is translation process, is very less, it is very much accurate, it is 99.9% accurate, but still, if there is a change in one amino acid even, if the protein is made up of multiple small subunits and if the protein is made up of single large polypeptide subunit. In this single large polypeptide subunit, if one amino acid, even the one amino acid is damaged, it is very prominent and it will affect the function. Whereas in case of small multiple polypeptides, if in any one subunit, one amino acid is damaged, now you tell me which damage is more prominent. Definitely the damage in the large polypeptide chain because it has only one active site. So even a one amino acid replacement can change the overall 3D structure of that polypeptide chain, making the protein inefficient. Whereas change in single subunit can be tolerated. So therefore, this limits the size of a protein and making the protein of multiple subunits. Therefore, the most protein with molecular weight greater than 1,000 have multiple subunits, identical or different, but they will have a multiple subunits. Now, one question from my end for you all, that is, what type of rotational symmetry are possible for a protein with 4 and 6 identical subunits? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.